So electron transport chain. We all know the utilization of this electron transport chain. By this whole transport system, we produce our ATP. So first of all, we should know about some of the terminologies. Acha, what is oxidation? It is the oxidation is the loss of electrons. And what is reduction? Gain of electrons, right? Now, there are some compounds which can act both in the form of oxidation compound and reduction compound. They are called redox couples. Redox couples. They can act both like oxidation compounds and reduction compounds. That means they can also lose electrons and they can accept electrons. Examples: NAD, NADH. We have it from pyruvate cycle and the carbohydrate metabolism. Then we have FAD, FADH. Okay. Now there is another term called redox potential. So redox potential is the capability of any redox couple to transport electrons. Okay, clear? It's the ability of redox couples to transport electrons. Clear? Are you clear about all those four terms? Okay. Now see, how many complexes are there in electron transport chain? 5. See, they are arranged. Okay. What is the location of this electron transport chain? Inner mitochondrial matrix. Okay. For example, like this is your inner mitochondrial matrix. Okay. All the complexes will be arranged like this: complex 1, complex 2, complex 3, complex 4, and complex 5. Up to this part, that is complex 1 to complex 4, we call the main electron transport chain, and this is the oxidative phosphorylation. This is the ATP synthesis. ATP is synthesized through this pathway. Okay. Now I will be explaining each of the complexes first, then I will make you understand how electrons are transported, how H plus is transported, and they form ATP. Okay. So we will start with complex 1. See, again the location will be inner mitochondrial matrix. If this is the inner mitochondrial matrix, this is the complex 1. See, we need portals, right? For everything, if we want to enter something, we need a portal for that entry. Complex 1 acts as a portal. It acts as a portal for NADH. Again, it is a redox couple, so it will get converted into NAD. And one H plus will flow out from inner mitochondrial complex to the outer part. Okay, so what is happening? This NADH, it is getting oxidized. Clear? Now see, when H plus ions are H plus ions are proton ions, right? These proton ions are pumping out of this complex 1. Some of the electrons, they are transporting from this complex 1 to a mobile electron transporter called ubiquinone. Now, what is mobile electron transporter? These are the mobile carriers. Like they will not affect, they will not be in your inner mitochondrial matrix. They will be outside of the inner mitochondrial matrix, and they will just simply carry electrons from one complex to other complex. You will understand the whole process, but first you should understand about the complexes. Okay? Now see, after creating these H plus ions, electrons are generated, and these electrons will be transferred to this factor Q or ubiquinone factor. Okay, this complex will have all the electrons from the complex 1, and again this COQ, it will transfer them to complex 2. We will come to complex 2 later. Okay, so what is happening? This complex 1, this NADH, it's getting oxidized, right? And someone is receiving those electrons, then it's reduced. So what can we name them? NADH, COQ, oxido, reductase. Clear? Are you clear about the naming? Okay. So we are done with complex 1. Now let's move to complex 2. Complex 2, it will also act as a portal. Now complex 1 was the portal for NADH. This one, complex 2, will act as a portal for FADH. And again, FADH, it's a redox couple. It will be converted into FAD, and electrons will be generated. Right? Are you getting my point? Are you getting all those? You are not. Okay, see. Are you not even clear about complex 1? This one. Similar to complex 1, it is also a portal. Now complex 1 was a portal for NADH. It will act as a portal for FADH. Okay? FADH will be converted into FAD, and electrons will be generated. Okay? What was happening from complex 1? We were having some electrons that were transferred into COQ. Right? Those electrons will again enter into complex 2. Okay? You're clear up to this point? I will make you understand how the whole electron transport chain is working. But first, you need to understand about the complexes. Okay. Now all the electrons, they are in complex 2. Right? There is a common reaction in the carbohydrate metabolism. Succinate is converted into fumarate by succinate dehydrogenase. This is happening in complex 2. Okay? So what can we name it? Succinate is there. And again, it will giving all the electrons to COQ. Right? It's ubiquinone. So can we name it succinate, ubiquinone, oxido, reductase? Correct? We can name it, right? Are you clear about the naming? Okay. After complex 2, all the electrons again will go to the mobile carrier, COQ. Okay? Now from COQ, it will be transferred to complex 3. Bear with me. You will get to know how the whole system is running. Okay? So complex 3. So after complex 2, all the electrons were in COQ. From COQ, it will enter into complex 3. Correct? Now complex 3 don't have any portal. Okay? All the electrons that were generating from 1 and 2 will be transferred to complex 3. Okay? Now see what is happening. This complex 3, it will give all those electrons to another mobile carrier called cytochrome C. And again, this cytochrome C will transfer all the electrons to complex 4. So this is receiving all the electrons, and this complex 4, it is the AND gate. Okay, it is the AND gate. It will oxidize all the electrons it will have. So can we name it cytochrome C reductase? Because this is receiving all the electrons and this is oxidizing all of them. So can we name it cytochrome C oxidase? Right? Up to this point, are you clear? There is an important fact. Copper is a compound of complex 4. We have a compound called copper in complex 4. Rest of the complexes, it's just heme based complexes, that is FeS, iron sulfate. Okay, clear? Now see how the whole process is running. Let me draw the inner mitochondrial matrix. This is the inner mitochondrial matrix. The first complex, what we had, what was the portal? What was the portal for the first complex? NADH. It will be converted into NAD, right? Now complex 2, what was the portal for? FADH, and it will be converted to FAD. Then we have our complex 3. And lastly we will talk about complex 5 lastly, in the last. Complex 4. Its name was cytochrome C reductase and its name was oxidation. See what is happening. As I have told you, when this will be converted into NAD, it will release H plus ions. So H plus ions are released and some electrons are generated. Okay, now these electrons, they will go to COQ, right? So COQ will have all the electrons as of now. Now there is a question, how many H plus are generated? 4 amount of H plus are generated in the first portal. Okay, now this COQ, it will have all the electrons, right? After this, it will transfer all the electrons to complex 2. And complex 2, by this reaction, will again generate some of more electrons. Now all the electrons, they will be again transferred to another mobile carrier, that is COQ. Now after this step, we have electrons that are transferred to COQ. All the electrons. Now all those electrons, they will be again transported to complex 4. Are you getting my point? How the electrons are transported? Okay, now complex 3 will have all the electrons which are generated up to this point. In this, there will be again a proton pump. Again 4 hydrogen ions will move from the inner mitochondrial matrix to the outside. Okay? So electrons are generated here, they were transported to COQ. Again electrons are generated here, they were also transported to COQ. Like all the electrons are flowing in this direction. Now complex 3 has all the electrons. This will be finally transferred to complex 4. In complex 4 we have a reaction. Half oxygen plus hydrogen giving H2O. Correct? This reaction, this whole reaction, it is triggered by the electrons that are being transported. That's why water is the end product of ETC. Are you getting my point? That's how we have water in the residual product of ETC. Now see, again 2 hydrogen ions, they will be pumped out of complex 4. Now we have 4 hydrogen ions from here, 4 hydrogen ions from here and 2 hydrogen ions from here. These all will be transported to the complex 5. Clear? Now after this transport, like 4, 4, 2, like 10 hydrogen ions are entering. they will be forming, they will be supporting this reaction. ADP plus Pi will be converted into ATP, and this whole reaction, it will be triggered by the hydrogen ions that are like carrying from all those complexes. ठीक है? अब देखो, when we have this NADH portal, how many hydrogen ions are being produced? 4, 4, 2. That means 10. For each of 4 hydrogen ions, we have 1 ATP. ठीक है? So how many ATPs are we getting? 2.5. So can we say this thing that if NADH is running the whole cycle, it will produce 2.5 ATP in total? And now see, now remove this NADH part. Now just think, always remember, both these cycles will not act together. Either this will be the portal or this complex 2 will be the portal. Okay, now see, remove this NADH, start from FADH. Now how many ATPs you are getting? 4, and 4 hydrogen ions you are getting, 4 and 2, that is 6. Then 1.5 ATP is being produced. Now you get this point that from 1 FADH we get 1.5 ATP, and from 1 NADH we get 2.5 ATP. Clear?